Welcome to the Deep Dive, where we plunge into the most groundbreaking discoveries and fascinating ideas shaping our world. Today, we're grappling with a question that sounds like it's ripped straight from the pages of science fiction.
Right.
What if we could genuinely rewrite the very blueprint of life itself? For centuries, this idea was confined to our wildest imaginings, a distant dream, but a profound biological discovery has now opened up possibilities that were once unthinkable, moving from the realm of fantasy to the very real.
And it's a reality that has evolved incredibly quickly. Genetic engineering, it has indeed come a remarkably long way from its early, often quite crude methods of gene modification.
Right.
We've seen a steady, deliberate march toward greater precision and accessibility. But what we're talking about today isn't just another incremental step forward in that progression.
No.
No, it's more like a monumental quantum leap. It has truly changed everything we thought possible in manipulating life.
That quantum leap, that game changer, is what we're calling CRISPR, the clustered, regularly interspaced short palindromic repeats.
Quite a mouthful.
It is, and it's a technology that emerged from one of the most unexpected, almost mundane places in nature, fundamentally transforming our ability to edit genomes. But, and this is really crucial for our deep dive today, with this immense power to solve some of humanity's most pressing global challenges comes significant technical perils and, well, some of the most profound ethical dilemmas humanity's ever faced.
Yeah, it's vital to grasp that CRISPR isn't merely a powerful tool to be wielded. It's an entire biological system that we've learned to repurpose.
A whole system.
Exactly. And its intrinsic revolutionary impact is dual-natured. It simultaneously offers incredible positive potential and introduces a really complex array of challenges that demand not just our attention, but our deepest consideration and wisdom.
And that dual nature, that incredible power balanced against profound peril, is exactly why this deep dive is so crucial. Our mission for you, our dedicated listener, is to unpack this incredibly complex technology. We're going to trace its surprising journey from its molecular origins in the microscopic world of bacteria to its incredible, truly life-changing applications in medicine, agriculture, and industry. And then we'll squarely confront the crucial dangers and weighty questions it presents. By the end of this deep dive, you'll have a clear, well-informed understanding of CRISPR, all the surprising facts and key insights, without feeling overwhelmed by information overload. So let's begin. The origin story of CRISPR is one of those truly fantastic tales of scientific serendipity
Absolutely.
and just persistent curiosity that reminds us how unexpected breakthroughs can be. It begins not in some sophisticated human lab filled with advanced equipment, but deep within the humble genome of E. coli bacteria.
Right, E. coli, of all places.
That's where these peculiar repetitive DNA sequences were first observed way back in 1987. They existed there largely unexplained for years, a sort of genetic enigma.
That's right. For a long time, they were merely an oddity, just a series of these repetitive DNA sequences separated by unique spacer segments within the bacterial genome. Didn't know what they did.
So when did that change?
Well, the crucial revelation, the moment the mystery really started to unravel, didn't fully come until around 2007, though foundational research by scientists like Francisco Mojica in the 1990s had already laid much of the groundwork.
Okay.
It was then that scientists finally elucidated their true function. They discovered that CRISPR actually functions as an adaptive hereditary defense mechanism against invading viruses in bacteria.
An immune system for bacteria.
Essentially, yes. Think of it as a kind of sophisticated molecular memory or an immune system for these tiny prokaryotic organisms, allowing them to remember and fight off past infections. It's an incredibly elegant natural defense system.
It's incredible to think of these tiny sequences existing, largely unnoticed in their full capacity for so long. What was the pivotal moment when scientists realized this bacterial quirk wasn't just a curiosity, but a potential revolution?
The true inflection point for modern genetic engineering, the moment this curiosity transformed into a powerful tool, arrived in 2012.
2012, okay.
That's when Emmanuel Charpentier and Jennifer Doudna, with their teams, published their landmark paper. They demonstrated that this natural CRISPR-Cas9 system, which originally defends bacteria, could be reprogrammed for targeted DNA cleavage in vitro.
In vitro meaning?
Essentially, outside of a living cell in a test tube. This breakthrough, which deservedly earned them the 2020 Nobel Prize in Chemistry, was the critical moment.
That was the spark.
Exactly. It was when an elegant natural system was transformed into a versatile, programmable tool for us to manipulate the genomes of other cells, including the far more complex eukaryotic cells, which encompass everything from plants to humans. It was, quite simply, a game changer because it showed we could wield nature's own tools.
To truly appreciate how we harness this, we first need to understand the nuts and bolts of that natural bacterial defense system. How does it operate in its original context before we human scientists got our hands on it?
Right. In its natural form, it operates in three distinct phases: adaptation, CRISPR RNA or cRNA biogenesis, and interference.
Okay, three phases.
During the adaptation phase, when a bacterium is attacked by a virus, it doesn't just succumb, it actually snips off a short fragment of the invading viral DNA.
Takes a sample.
Precisely. This fragment, which scientists call a protospacer, is essentially a tiny piece of the viral invader's genetic fingerprint. It's then integrated into the bacterium's own CRISPR array, creating what we can think of as a molecular mugshot.
A mugshot, I like that.
Or a memory of that specific viral threat. This genetic library of past infections allows the bacteria to recognize subsequent attacks from the same virus.
So it learns.
It learns. Then, next, during eRNA biogenesis, this stored information, these mugshots are processed into functional guide RNAs.
Guides.
Finally, in the interference phase, these guide RNAs then direct a Cas protein, which acts like a molecular scissor, to precisely cleave and neutralize the foreign DNA upon re-exposure to that specific virus. It's a beautifully choreographed immune response at a molecular level.
It sounds incredibly complex, this bacterial defense system, a true marvel of natural selection. How did scientists ever make the leap from observing this in bacteria to realizing it can be repurposed as a universal gene editing tool for us?
Well, we essentially streamlined that intricate natural defense system, distilling its core functionality for our own gene editing goals.
Oh.
The most widely adopted system, CRISPR-Cas9, centers on two core components.
Okay, what are they?
The Cas9 nuclease, which is the actual molecular scissor, and a single guide RNA, or sgRNA.
sgRNA.
Yes. This sgRNA is a synthetic fusion of two native RNA components found in the bacterial system, the crRNA, and something called a TREC RNA. The sgRNA is absolutely critical because it acts like a GPS for the Cas9 protein.
Yeah, the GPS analogy.
Exactly. Its 20-base pair sequence is designed to precisely match the target DNA sequence we want to edit, guiding the Cas9 protein right to that specific genomic spot with incredible accuracy. And there's one more crucial detail, a molecular gatekeeper, if you will. The protospacer adjacent motif, or PAM sequence. PAM. PAM. This is a short, specific DNA sequence that must be located immediately downstream of the target site that the sgRNA is trying to bind to.
So it's like a docking requirement.
Precisely. Cas9 simply cannot bind to the target DNA and cleave it without first recognizing and engaging with this PAM sequence. Once the sgRNA guides the Cas9 to the correct location and the PAM is recognized, the Cas9 enzyme undergoes a conformational change.
It changes shape.
It does. Its two specific cutting sites, imagine them like a perfectly aligned pair of molecular scissors, then precisely cleave both strands of the DNA, creating what we call a double-strand break, or DSB.
The double-strand break, that precise cut in the DNA, that's the key moment, right?
Yeah.
That's where the cell itself becomes an active and essential participant in our editing process.
Exactly. The cell's natural DNA repair machinery, which is constantly at work maintaining the integrity of our genetic material, immediately kicks into high gear to fix it.
So we break it, the cell fixes it.
We're essentially leveraging the cell's own sophisticated repair crew to do a lot of the heavy lifting for us based on how we want the fix to happen.
How does that work? How do we influence the fix?
Well, the cell has two primary pathways for repairing these double-strand breaks, and we cleverly exploit both of them to achieve different editing outcomes.
Okay, pathway one.
The first is non-homologous end joining, or NHEJ. Think of this as the cell's rapid, often error-prone, quick-fix pathway.
Quick, but maybe messy.
Exactly. It's like patching a broken pipe in a hurry. It simply ligates the broken DNA ends back together, but in doing so, it frequently introduces small insertions or deletions, what we call indels, at the cut site.
Indels.
Right. These indels can disrupt the gene's reading frame, effectively turning the gene off and leading to a functional gene knockout, or KO. This is our go-to method for silencing a gene.
So break it, let NHEJ fix it badly, and the gene stops working. Got it. What's the other pathway?
The second pathway is homology-directed repair, or HDR. This is the cell's high-fidelity, precise repair pathway.
High fidelity. Sounds better.
It is, for precision work. Unlike NHEJ, HDR requires a homologous donor DNA template, essentially, a blueprint, a piece of DNA that shares sequence similarity with the region around the break.
And we provide that template.
As researchers, yes, we can provide this template. By designing it with a desired new sequence, we can leverage HDR to insert new genetic material, correct a specific mutation with pinpoint accuracy, or essentially swap out an old sequence for a new one. This process is known as gene knock-in, or KI.
Knock-in.
This is how we introduce specific changes or entirely new genetic information. Precisely.
So when we put this all together, what does this actually mean for you, the listener, in terms of what's now possible? It sounds like we can direct this molecular scissor to a specific location, and then simply by choosing which repair pathway the cell is encouraged to use, or by providing that template, we can either turn a gene completely off,
with NHEJ usually,
or we can precisely change it, inserting new information or correcting a single base pair using HDR.
That's the core idea.
All of this is done by cleverly harnessing the cell's own sophisticated repair mechanisms, almost like having a tiny, incredibly accurate genetic surgeon. But then the patient, the cell itself, performed its own recovery and reconstruction based on our precise instructions.
A good analogy. We make the cut. The cell does the edit based on the context we create.
Now, it's worth noting, as you mentioned, CRISPR wasn't the first gene editing tool to come along.
No, definitely not.
But it very quickly became the dominant one, effectively eclipsing its predecessors with remarkable speed. So, what made CRISPR so revolutionary that it soared past earlier? Complex technologies like zinc finger nucleases, or ZFNs.
ZFNs, yes.
And transcription activator-like effector nucleases known as TALENs.
TALENs, right.
Why did CRISPR fundamentally democratize this field?
That's a critical question, and the answer lies squarely in its stark advantages, particularly its inherent simplicity and cost-effectiveness.
Simplicity and cost. Okay.
Let's look at a quick comparison, drawing from the source material. With CRISPR-Cas9, the mechanism of targeting is based on RNA-DNA base pairing. This is incredibly straightforward. You simply design a short RNA sequence that precisely matches your target DNA.
Like a key fitting a lock.
It's like knowing exactly which key fits which lock, because you're working with a universal, predictable genetic code. Very easy to design that RNA guide.
And the others? ZFNs and TALENs.
Contrast that with TALENs and ZFNs. Their targeting mechanism relies on complex protein DNA binding. This is far more intricate because you have to engineer entirely new proteins for each specific target DNA sequence.
Oh, a whole new protein tool for every single job.
Pretty much. For TALENs, each protein domain recognizes a single nucleotide, and for ZFNs, each domain recognizes a 3-base pair triplet. Designing and assembling these bespoke proteins is a laborious, highly specialized, and often unpredictable process, demanding immense expertise.
Right, that sounds way harder.
It is, and this brings us directly to design simplicity. CRISPR requires only that single guide RNA, the sgRNA, which is remarkably easy to design and synthesize in any molecular biology lab. You simply input your desired 20-base pair sequence, and you've basically got your guide.
Hug and play, almost.
Almost. For TALENs and ZFNs, you're essentially constructing complex multi-domain proteins from scratch for every new target. This is not just challenging, it's a significant barrier in terms of time, specialized expertise, and resources.
Which must affect the cost.
Then there's the monumental difference in cost. CRISPR is comparatively inexpensive. The guide RNAs are cheap to produce, and the Cas9 enzyme is now readily available.
Okay.
Its predecessors, TALENs and ZFNs, were prohibitively expensive and time-consuming precisely because of the bespoke protein engineering required. This dramatic cost difference alone massively impacted accessibility and truly democratized the field.
Made it available to way more labs.
Absolutely. And then in terms of efficiency, CRISPR is highly efficient and rapid, delivering precise edits quickly and reliably. TALENs were less efficient, and ZFNs were often the least efficient among the three.
So fast results with CRISPR, too.
Generally, yes. This directly affects how quickly and reliably researchers can achieve their desired genetic modifications, which translates into faster scientific discovery and faster development of applications.
And can you do multiple edits at once?
That's another key advantage. Multiplex editing, or the ability to target multiple genes simultaneously is quite feasible and routine with CRISPR. You simply introduce multiple sgRNAs, each targeting a different gene, and the Cas9 enzyme can work on them concurrently.
Wow.
For TALENs and ZFNs, multiplexing is far more difficult and much less efficient, again due to the need to engineer separate, complex protein systems for each target.
So this comparison truly highlights a fundamental reason for CRISPR's rapid ascent. Its power isn't just about what it can do, but how easily, quickly, and affordably it can do it.
That sums it up well.
It's a direct consequence of its inherent simplicity and cost-effectiveness. This democratized genome editing, taking it out of the hands of a few highly specialized labs and making it accessible to virtually any research institution with basic molecular biology capabilities.
It spread like wildfire through the research community.
This unprecedented accessibility has accelerated discovery and application at an astonishing pace.
Wow.
But you mentioned earlier that this also creates a new set of challenges in governance and oversight. Is it because so many more people can now do this work with relative ease?
Precisely. The widespread accessibility is a double-edged sword. On one hand, it fosters incredible innovation globally, fantastic progress. On the other, it means regulatory bodies and ethical frameworks struggle to keep pace with a technology that's no longer confined to a few elite labs, but potentially accessible to almost anyone with a lab bench and the basic reagents.
Right.
This broad reach elevates the urgency of the ethical and safety discussions we'll definitely need to get into later.
Indeed. But first, let's talk about the power side of that equation. The promise of CRISPR in medicine is no longer theoretical, right? It's a clinical reality that is already changing lives, moving from lab benches to patient bedsides at a truly astonishing speed.
Absolutely. The landmark achievement here is the FDA approval in December 2023 of Casgevy, or exagglogen autotemcel. You might have heard it called Exacel before approval.
Casgevy, okay.
This is the first CRISPR-based therapy approved for clinical use in the United States, specifically for Sickle Cell Disease, or SCD, and transfusion-dependent beta thalassemia, TDT.
Two really tough genetic blood disorders.
Exactly. This represents a monumental shift in how we approach their treatment, moving from just managing symptoms to offering a functional cure.
What does this actually mean for patients living with these debilitating conditions? How does Casgevy work inside the body to bring about such a profound change?
Okay, so Casgevy works by first taking a patient's own hematopoietic stem and progenitor cells. These are essentially blood-forming stem cells out of their body.
Taking their own cells out.
Yes. These cells are then edited ex vivo, meaning outside the body, using CRISPR-Cas9 technology to precisely target and modify a specific genetic switch.
Switch.
Yeah, a switch that normally turns off fetal hemoglobin production after birth. The edit basically flips that switch back on, increasing the production of fetal hemoglobin, which is a type of hemoglobin naturally produced before birth.
And fetal hemoglobin works better than the faulty adult kind in these diseases.
Exactly. Fetal hemoglobin then compensates for the defective adult hemoglobin that causes the painful and life-threatening symptoms of sickle cell disease and beta thalassemia. Once edited and confirmed, these corrected cells are reinfused into the patient.
After some conditioning therapy, right?
Yeah.
Like chemo.
Yes, that's an important part of the process to make room for the edited cells. Then they're reinfused, essentially giving the patient a new functional blood system derived from their own edited cells.
And the clinical trial results have been nothing short of remarkable. I saw some numbers. 96.7% free of crises, 100% hospitalization free. Those aren't just statistics. Those are profound changes in human lives.
They really are transformative. In the pivotal study, an astounding 96.7%, that's 29 out of 30 evaluated patients, were completely free of vaso-occlusive crises, or VOCs, for at least 12 consecutive months following treatment.
Those are the incredibly painful episodes.
Yes, the excruciatingly painful episodes that characterize sickle cell disease and lead to organ damage. Even more significantly, 100% of these patients remain hospitalization-free for the same duration.
Wow.
Beyond these critical disease markers, patients also reported clinically meaningful and sustained improvements in their overall quality of life, spanning physical, social, and emotional well-being.
That's huge.
These outcomes truly underscore CRISPR's potential to offer a one-time functional cure, essentially rewriting the disease narrative for those affected by these devastating genetic diseases. The speed to approval for such a complex therapy is indeed quite remarkable, signaling the immense promise the technology holds.
And it's not just monogenic disorders. Those diseases caused by a single gene mutation like sickle cell. CRISPR is also revolutionizing oncology, particularly in enhancing immuno-oncology and CAR T-cell therapies, which have already shown incredible promise in fighting certain cancers.
That's correct. Traditional CAR T-cell therapies are autologous, meaning they use a patient's own T-cells, which are then modified and reinfused. This process is highly personalized, but also incredibly costly and time-consuming.
Right, takes weeks or months sometimes.
Exactly. CRISPR is enabling the generation of off-the-shelf allogeneic CAR cells.
Off the shelf, meaning
This means we can create a standardized product from healthy donors that can be readily available to many patients, addressing many of the logistical and manufacturing challenges of the autologous approach, much faster, potentially cheaper.
how does CRISPR make that possible? Doesn't the patient's body reject donor cells?
That's where the editing comes in. Critically, researchers use multiplex gene editing with CRISPR, meaning they make several gene modifications simultaneously in those donor T cells.
Multiple edits at once. Okay, what kind of edits?
So to simplify, you're essentially building a super T cell that's more aggressive against the cancer and simultaneously making it invisible to the patient's own immune system to prevent rejection. That's the dual goal.
Ah, I see. A two-pronged attack.
Exactly. We're giving these T cells a dual boost. We knock out genes that act like brakes on the T cells, such as RAGNS1 and TGFBR2, to significantly enhance their potency and persistence against tumors, making them more aggressive and longer lasting.
Kicking the brakes off.
Right. And at the same time, we knock out genes like MHCI and the T-cell receptor, or TCR, to make these donor cells invisible to the patient's immune system.
The invisibility cloak.
Pretty much. This prevents the patient's body from recognizing them as foreign and rejecting them, allowing for a more powerful, persistent, and universally applicable anti-tumor cell therapy. It's a sophisticated act of biological engineering.
Beyond cancer, CRISPR also holds immense promise for infectious diseases, which is, of course, a critical area for global health, especially with emerging pathogens.
Indeed, we're seeing two key applications here. First, as a powerful and rapid diagnostic tool.
Diagnostics, how does that work?
Platforms like Sherlock, developed by Mammoth Biosciences and Feng Zhang's team, use CRISPR components, specifically different Caucase enzymes like Cas13, to detect specific genetic sequences like viral RNA.
So it can find the virus's genetic signature.
Exactly. It can rapidly detect viral RNA from pathogens such as COVID-19, Ebola, and Zika virus. This offers a faster, more portable, and often more cost-effective alternative to traditional methods like PCR.
Faster than PCR, that's impressive.
It can be, and often doesn't require the same lab equipment, making it crucial for rapid outbreak response and surveillance, particularly in resource-limited settings where quick identification can save lives.
Okay, diagnostics is one. What's the other?
Second, CRISPR is being explored as a direct therapeutic to combat chronic viral infections. The idea here is quite bold, to precisely excise integrated viral DNA from the host genome.
Cut the virus right out of our DNA.
Essentially, yes, cutting out the infection at its source. We've already seen compelling proof-of-concept studies demonstrating its success in removing HIV DNA from infected cells in lab settings.
Oh, HIV.
Yes. This offers a potential path to a functional cure for persistent viral infections that currently require lifelong treatment, shifting the paradigm from management to eradication. Still early days for therapies, but incredibly promising.
Absolutely. Moving from human health, the global challenges of food security, climate change, and sustainability demand innovative solutions. It's truly inspiring to see how CRISPR is stepping up in agriculture to meet these pressing needs. It's not just about human health, but the health of our planet and its ability to feed us.
It truly is a testament to its versatility. In crop science, CRISPR is being used extensively to engineer crops with enhanced abiotic stress tolerance.
Abiotic stress, meaning things like drought and heat.
Exactly. Making plants more resilient to environmental challenges like drought and heat, which are becoming increasingly prevalent due to climate change. For instance, in maize, researchers have precisely modified the ZMHDT103 gene, showing significantly improved drought tolerance without compromising overall growth or yield.
So tougher corn.
Tougher corn, yes. In wheat, editing the TARTK1 gene has led to enhanced water absorption, a crucial trait in arid and semi-arid regions. Another innovative approach involves manipulating SAL1 genes to increase the production of osmoprotectants like proline.
Osmoprotectants.
They're molecules that help protect cells from dehydration. So increasing them helps plants withstand dry periods more effectively by protecting their cells and maintaining cellular function. These are crucial adaptations for future food security.
Makes sense. What about diseases? Pests?
Beyond climate resilience, CRISPR is instrumental in creating crops that are naturally resistant to diseases. This can significantly reduce our reliance on chemical pesticides and herbicides, making agriculture more sustainable.
How do they do that? Edit resistance in.
It's achieved by precisely editing host resistance R genes or susceptibility S genes. By disabling a susceptibility gene, the plant can no longer be infected by a particular pathogen. The pathogen sort of loses its entry key.
Ah, lock the door.
Exactly. Alternatively, incorporating a resistance trait from another variety using CRISPR can create durable, broad-spectrum resistance against viruses, fungi, and bacteria. This not only improves yields in farmers' livelihoods, but also makes agriculture more environmentally friendly by reducing chemical inputs.
So, from making crops more resilient and productive, it's clear CRISPR is transforming agriculture. But the benefits don't stop there, do they? How is this technology extending its reach into animal agriculture and livestock production?
In livestock, CRISPR offers a powerful means to improve animal welfare, enhance disease resistance, and increase overall efficiency in food production, all crucial for meeting global demand ethically.
Animal welfare is a big one.
It is. For example, scientists have successfully used CRISPR to make pigs genetically resistant to the devastating porcine reproductive and respiratory syndrome virus, or PRRS.
PRS, I've heard of that,
Right.
causes huge losses.
Immense economic losses globally, yes. And it often necessitates extensive antibiotic use to mitigate secondary infections. By making pigs inherently resistant, we can significantly reduce both livestock losses and our reliance on antibiotics in animal farming, which helps address concerns about antibiotic resistance in humans, too.
That's a major benefit.
Yeah.
What about direct welfare, like the horns?
Yes, exactly. The technology can address animal welfare concerns directly. For instance, eliminating horn development in cattle through gene editing avoids the need for painful physical dehorning procedures later on.
So hornless cattle from birth.
Right. It improves the lives of the animals and enhances the safety of farm workers handling them. These are targeted, humane interventions made possible by precise gene editing.
Okay, so beyond these direct applications we've discussed, medicine, agriculture, livestock, CRISPR is proving to be an absolute powerhouse for fundamental research and in the fascinating world of making new products through bioproduction. It's not just about cutting and pasting genetic code. It's about incredible precision and control over biological processes.
That's a critical point for understanding its broader impact. One of the most exciting advancements here is the use of a catalytically inactive Cas9, or dCas9.
dCas9, the DEED stands for.
Dead Cas9, essentially. This variant of Cas9 is ingeniously engineered so it can still bind to DNA, guided by its sgRNA, but crucially, it cannot cut the DNA.
So it finds the spot but doesn't cut. What's the point then?
It acts purely as a programmable scout, or maybe like a roadblock or a booster. It guides the complex to a gene's promoter region without causing a double-strand break. This allows for incredibly precise, programmable gene regulation, turning genes on or off at will without altering the underlying sequence.
Controlling gene expression, not the gene itself.
Precisely. We can leverage this in two powerful ways. In CRISPR interference, CRISPR, dCas9 is used to physically block RNA polymerase from transcribing a gene, effectively repressing or silencing gene expression.
Like putting a roadblock in front of the gene's on switch.
Exactly. It's like putting a roadblock in front of a factory's production line. And conversely, in CRISPR activation, CRISPR, dHEAS9 is fused to transcriptional activators.
Things that turn genes on.
Right. It recruits them to a gene's promoter and significantly boosts its expression. This is like adding a turbo boost to that factory's production.
These tools sound incredibly valuable in metabolic engineering, where you're essentially trying to get cells, like yeast or bacteria, to produce specific compounds more efficiently.
Precisely. In metabolic engineering, this reversible, programmable control is invaluable. Researchers can use CRISPR and CRISPR to meticulously redirect metabolic flux, essentially, fine-tuning the cell's internal biochemical pathways.
Rerouting the cell's chemistry set.
You could put it that way. To enhance bioproduction yields in industrially relevant microorganisms like E. coli and S. cerevisiae. For example, you might want to increase the output of a specific chemical, a biofuel, or a pharmaceutical compound. The ability to rapidly and reversibly control the expression of multiple genes simultaneously without permanent genomic modification offers an immensely flexible and powerful platform for optimizing complex biological pathways in a way that was previously unimaginable. It's about engineering life at a truly fundamental level for industrial benefit.
Okay, we've talked a lot about the power. Now we need to pivot to the peril. This powerful tool, for all its immense potential, also carries significant technical dangers. These aren't just abstract risks. They're rooted in the very mechanism of how CRISPR makes that double-strand break in our DNA.
Yes.
It's a core danger that we absolutely need to confront, especially when considering its use in human therapies.
You're absolutely right. The primary technical peril of traditional CRISPR-Cas9 is the risk of unintended mutations, which can have profound consequences. These manifest in two major forms.
Two main types of errors.
Yes, off-target effects and, more broadly, a potential for genome-wide instability. Off-target effects occur when the Cas9 enzyme, guided by its sgRNA, cleaves DNA at unintended sites within the genome.
Cutting in the wrong place.
Exactly. These unintended sites might not be perfectly identical to our target, but they have a similar enough sequence. Cas9 can, in fact, tolerate a certain degree of mismatch in the target DNA sequence, especially outside a critical area known as the seed region closest to the PAM.
So it's not always perfectly specific.
It's highly specific, but not absolutely perfect, especially in a genome as vast and complex as the human genome. Similar or homologous sequences are ubiquitous, making the risk of off-target cutting a persistent and significant concern.
Okay, that's off target. What's the other issue?
Moreover, even at the intended target site, the error-prone nature of the non-homologous end joining, or NHEJ, repair pathway, that quick fix we talked about, can lead to a variety of on-target mutations that we didn't intend.
So even if it cuts in the right place, the repair can mess things up.
It can introduce random small insertions or deletions, yes. But a potentially more profound concern is the possibility of unexpected genome-wide instability affecting parts of the genome far from our target.
Instability across the whole genome.
There's evidence suggesting this can happen. A study analyzing CRISPR-Cas9 edited Drosophila fruit flies using whole genome sequencing revealed numerous unintended mutations, including single nucleotide variants, SNVs, insertions, and deletions, indels, distributed across the genome.
Wow, all over the place.
Yes, and while the majority of these were found in non-coding regions, the so-called junk DNA, and didn't directly affect the organism's visible traits, their mere existence highlights a potential danger to the integrity of the genome as a whole.
So integrity of the genome as a whole, that sounds like a fundamental biological principle. What could subtle long-term instability, even in non-coding regions, mean for an organism's health or evolution decades down the line, beyond the immediate risks like cancer?
That's precisely the deeper, more unsettling question. Even subtle long-term instability in non-coding regions, which we now know are often involved in gene regulation or maintaining the structural integrity of chromosomes.
They're not just junk.
Not at all. Damage there could have unforeseen cascading effects. It might subtly alter gene expression patterns over time, increase susceptibility to other diseases later in life, or even affect reproductive fitness in ways we don't immediately detect.
A delayed reaction.
Possibly. In an evolutionary sense, introducing such widespread, unpredictable changes could impact how an organism adapts or evolves, potentially creating entirely new vulnerabilities for future generations. It's the butterfly effect at a genetic level, where a small, seemingly benign change could lead to larger, unpredictable biological consequences down the line. It highlights the immense responsibility involved.
And in humans, getting back to therapies, what are the worst-case scenarios from these unintended mutations?
In a direct therapeutic context for humans, the most severe implications could be catastrophic. They could potentially disrupt essential genes, which could have serious adverse effects on normal cell function or lead to severe developmental issues. There's also the risk of triggering an adverse immune response in the patient, as the body might identify the edited cells as foreign or abnormal, leading to rejection or inflammation.
Right, the body attacking the therapy.
But perhaps most critically, there's the danger of inducing oncogenesis, that is, inadvertently promoting cancer.
Cancer. How?
By either accidentally activating an oncogene, a gene that drives uncontrolled cell proliferation, or by inhibiting a tumor suppressor gene, one that normally prevents cancer, thereby removing a critical cellular safeguard. These are the life and death stakes when we talk about unintended consequences in human gene therapy.
Oh, very sobering. The good news, though, if we can call it that, is that these very real dangers haven't brought innovation to a halt. Instead, they've spurred incredible creativity, leading to a new wave of even more precise and safer tools.
That's right.
It's a crucial cause and effect relationship in scientific progress, demonstrating how challenges drive ingenuity.
Absolutely. The fundamental limitation of traditional CRISPR-Cas9, its reliance on making that DNA double-strand break, has directly catalyzed the development of next-generation gene editing platforms. These new tools are specifically designed for superior precision and safety,
Making them better for medicine.
far better suited for sensitive applications like human therapy, where every base pair change counts and minimizing risk is paramount.
So what are these new tools?
Let's look at two of the most significant advancements: base editing and prime editing.
Base editing and prime editing. Okay.
Base editing addresses the double-strand break problem head-on by directly modifying a single nucleotide without ever breaking the DNA backbone entirely.
No double break.
How? This technology consists of a modified Cas9, specifically a Cas9 nick-Cas, remember dCas9, the dead one. A nick-Cas just nicks one DNA strand, not both, which is then fused to a deaminase enzyme, an enzyme that chemically changes one DNA base letter into another. The nickase opens a small window, and the deaminase goes in and makes the change. For instance, it can change a cytosine C to a thymine T, or an adenine A to a guanine G.
Swaps one letter for another chemically.
Exactly. This method is highly efficient and predictable for single base corrections, making it an ideal tool for correcting an estimated 30% of known disease-causing point mutations, which are these single-letter changes in the genetic code.
30% is a lot of genetic diseases. Okay, that's base editing. What about prime editing?
Prime editing represents an even more versatile search and replace technology that also elegantly avoids double-strand breaks.
Search and replace, like on a computer.
Kind of, yes. It's composed of a Cas9 nickase, again just nicking one strand, fused to a reverse transcriptase enzyme.
Reverse transcriptase, like viruses use.
Exactly, an enzyme that can write RNA sequence back into DNA.
No.
It's guided by a specialized prime editing guide RNA, or PEG-RNA. What's unique about the PEG-RNA is that it doesn't just direct the complex to the target site, it also carries the template for the new genetic information that we want to insert right there at the nick site.
So the guide RNA itself holds the correction.
Yes. Prime editing's unique mechanism allows it to make all 12 possible single-base substitutions and also small insertions and deletions with exceptional accuracy.
All 12 changes plus small fixes. That sounds incredibly versatile.
It is. And a key advantage of prime editing is its unusually high DNA specificity and remarkably low off-target rate. This is attributed to what are called its 3 edit checks, 3 successive hybridization events, like 3 security checkpoints, that must occur precisely for an edit to be completed.
Built-in safeguards.
Exactly, acting as built-in safeguards that ensure accuracy.
This sounds like a significant leap forward in precision. So what does this all mean for the future of gene editing, especially in sensitive areas like human therapy? How have these advancements truly shifted the landscape?
It signifies a clear and crucial evolution in our genetic toolkit, prioritizing safety and precision above all else. Let's compare them quickly.
Okay.
With traditional CRISPR-Cas9, a double-strand break is required. This allows for gene knockouts via NHEJ or precise knock-ins via HDR. However, as we discussed, it carries a significant off-target risk due to DNA mismatches and genome homology.
The blunt instrument.
Relatively speaking, yes, making it less ideal for precise therapeutic applications due to those inherent DSB-related risks. The cell's reliance on error-prone repair is a key issue.
Right. Then base editing.
Base editing, on the other hand, makes only a single-strand nick, elegantly avoiding the problematic double-strand break altogether.
Yeah.
This is critical because it bypasses the most damaging cellular repair pathway. It enables 4 types of point mutations, C to T, T to C, A to G, and G to A, with very high efficiency, especially for single-base corrections.
And less risk.
Its off-target risk is very low because it doesn't create the same level of genomic stress, and it's highly suitable for single-base corrections in a clinical context where precision is paramount.
And prime editing.
Prime editing also only requires a single-strand nick, again avoiding the double-strand break. Its versatility is remarkable. It's capable of making all 12 types of point mutations, plus small insertions and deletions.
The most flexible one.
Potentially, yes. While it might still require a bit more optimization for efficiency in some cases compared to base editing, its off-target risk is also very low due to its unique 3 hybridization checks, which greatly enhances its specificity and minimizes unintended edits.
So very safe too.
Extremely precise. This makes it highly versatile and exceptionally precise, with the theoretical potential to correct the vast majority, maybe up to 89%, of known pathogenic alleles that cause disease.
Wow, 89%. That's incredible potential. This progression really shows a clear movement from what was a powerful but relatively blunt tool to a suite of much more refined, specialized instruments,
Yes.
addressing a wider range of genetic errors with far greater control. These next-generation editors truly prioritize precision and safety, which is absolutely critical for sensitive applications like human gene therapy, where the stakes are quite literally life and death. It's an exciting demonstration of how science responds to its own limitations and pushes the boundaries of what's possible.
It's a field moving at incredible speed.
Okay, but beyond the technical challenges and these amazing solutions, CRISPR introduces some of the most profound ethical and societal dangers that humanity has ever grappled with. It challenges our fundamental concepts of medicine, justice, and even human identity itself.
This is where the conversation gets really complex.
It's a complex web of moral, social, and philosophical questions.
That's precisely why the debate on human germline editing is such a central point of contention, sparking intense ethical discussions globally. We need to understand the clear distinction here.
Okay, the distinction is key.
Somatic cell editing modifies a patient's non-reproductive cells to treat a disease. The changes are confined to that individual and are not passed on to future generations.
Like TSGI.
Exactly like KSJY for sickle cell disease. That falls squarely into this category, and there's broad scientific and public support for such therapeutic uses. The ethical framework here is largely aligned with traditional medical interventions treating disease in an individual.
Okay, that's somatic. What's germline?
In stark contrast, germline editing involves modifying the DNA of eggs, sperm, or early embryos. These changes, if successful, would be permanent and would be passed down to all future generations.
Inheritable changes.
Yes. This introduces a host of unresolved issues, biological, ethical, and societal, which is why there's an almost universal global moratorium on its clinical use today. It fundamentally alters the human gene pool, and the implications ripple across all of humanity, not just an individual.
This is where the common concern about a slippery slope comes in, isn't it? Even if the initial intentions might be noble, like curing a terrible inherited disease, in a way that prevents it for all descendants.
Which sounds good on the surface.
Right, but why are people so incredibly concerned that it could lead to something far more problematic?
The slippery slope fear is potent because it suggests that once we cross the threshold of using CRISPR for treating serious genetic diseases in the germline, it could inevitably lead to its use for non-therapeutic enhancement purposes.
Enhancement? You mean making people better?
That's the fear. This is the concept colloquially known as designer babies. The concern isn't just theoretical, it's rooted in profound fears about a potential return to a modern form of eugenics.
Eugenics, that's a heavy word.
It is. We could find ourselves in a society where genetic traits are selected not purely for health and disease prevention, but for enhancement perhaps, intelligence, physical characteristics, maybe even personality traits, though the genetics of those are incredibly complex. But the fear is it could lead to a genetically homogeneous society that values only certain characteristics, creating new forms of discrimination, deepening existing inequalities, and fundamentally altering our understanding of human diversity and dignity.
What are the core ethical principles at stake here for you, the listener?
Well, several really fundamental ones. For instance, the principle of informed consent is fundamentally challenged. The individuals who would be most affected by germline edits unborn future generations cannot possibly provide their consent to these irreversible heritable changes.
We're making choices for people who don't exist yet.
Exactly. We are making choices for them. Furthermore, many individuals and religious groups hold strong moral objections. They view the manipulation of human embryos and the fundamental building blocks of life as playing God, or as an unnatural act that interferes with human nature and dignity, crossing a line that should remain uncrossed. These are not easily dismissed concerns and are at the heart of the global moratorium.
Okay, so germline editing is a major ethical minefield. But even with somatic therapies, like Casgevy, the revolutionary power of CRISPR is linked to a very real danger of social inequality. We have these incredible life-changing therapies, but at what cost? And for whom?
Yes, this is a critical point that demands our immediate and sustained attention. Let's take the tangible example of Casgevy again, the CRISPR therapy for sickle cell disease.
The price tag.
The price tag. A single dose of this therapy is currently priced at $2.2 million in the U.S.
$2.2 million.
Right. Now, while this figure might, in some analyses, be demonstrably lower than the lifetime healthcare costs for managing transfusion-dependent beta thalassemia or severe sickle cell disease.
Which can also run into millions over a lifetime.
Correct. But that initial $2.2 million figure raises serious, immediate concerns about equitable access. If a cure exists, but only the ultra-wealthy or those with exceptional insurance coverage can afford it, we risk exacerbating existing health disparities.
Creating a genetic divide.
We risk creating a new genetically privileged class of individuals where access to fundamental health and well-being is determined by economic status.
And this problem isn't just about the steep price tag, is it? It runs much deeper than that, touching on historical biases and systemic inequalities. Are there any parallels in medical history here?
Absolutely, you're right. The issue is compounded by historical and systemic factors. We've seen similar patterns with other transformative therapies like early HIV treatments or certain expensive biologics for autoimmune diseases, where initial high costs severely limited access.
We've seen this before.
We have, and the lesson is clear. If we don't proactively address affordability and distribution from the start, the benefits remain concentrated among the privileged. This problem is further exacerbated by the historical underrepresentation of minority and socially disenfranchised groups in genomic research itself.
How does that affect things?
Without adequate representation in genetic databases and clinical trials, the therapies developed may be less effective or even unsafe for these populations simply because their genetic diversity wasn't adequately considered during development.
So the cure might not even work as well for everyone.
That's a real risk. This could significantly widen the already existing health equity gap, deepening social disadvantage. Addressing this requires a proactive, deliberate commitment to enhancing diversity in research, building genuine partnerships with underserved communities, and frankly, rethinking our entire healthcare economic model.
A huge challenge.
It is. We must ensure that the benefits of this groundbreaking technology are distributed equitably across all of humanity, rather than becoming another source of social division.
Okay, shifting focus again slightly. The power of CRISPR extends far beyond humans to our environment, introducing novel and very serious biosecurity risks that we are only just beginning to fully understand and appreciate.
Yes, this brings us to one of the most controversial and potentially dangerous applications in the environmental sphere: gene drives.
Gene drives, I've heard that term. What exactly is it?
A gene drive is a powerful genetic engineering technology, often built using CRISPR, that forces an edited gene to be inherited by all offspring, essentially bypassing the normal 50% chance of Mendelian inheritance.
It guarantees inheritance.
It strongly biases inheritance, yes.
Yes.
This ensures that a specific trait or gene spreads rapidly, maybe even exponentially, through an entire population within just a few generations.
Why would we want to do that?
Well, on the one hand, this tool holds immense potential benefits. The classic example is eradicating disease vectors, like the malaria-carrying mosquitoes that cause so much suffering and death globally. A gene drive could potentially spread infertility or disease resistance through the mosquito population, crashing it or making it harmless.
Wiping out malaria sounds amazing, but
However, the profound danger is that it also poses a potential existential threat to biodiversity and ecosystem stability. The risk lies in what our source calls technology escape.
escape, like it gets out of the lab.
Or gets out of the target population or region. Imagine an accidental or even intentional release of a gene drive into the environment. This could decimate an entire target species, which might be the goal in some cases, like with mosquitoes.
But what if it jumps species?
Yeah.
Or has unintended effects?
Exactly. It could disrupt ecosystems in unforeseen and unpredictable ways. The long-term ecological consequences are largely unknown and incredibly complex. Eliminating one species, like a specific mosquito, could remove a crucial food source for other species, leading to cascading effects throughout the food web.
Domino effects.
Right. It could also promote the proliferation of invasive pests by removing natural competitors or predators. We could create a complex, unpredictable chain reaction across entire ecosystems that could be irreversible. This isn't just about losing a single species, it's about potentially destabilizing entire biological communities.
And it seems like this rapid pace of CRISPR development, particularly with gene drives, has unfortunately outpaced our ability to regulate it effectively,
Just putting it mildly.
leaving us with this immense power, but without a clear global roadmap or safety net.
That's precisely the core problem.
Yeah.
There is a significant governance vacuum. The speed at which CRISPR technology has advanced, coupled with its increasing accessibility and its far-reaching implications, means that our regulatory frameworks and ethical guidelines have struggled immensely to keep pace.
We're playing catch-up.
We are definitely in uncharted territory. There is a critical and urgent need for a unified global framework for oversight and regulation. This framework must be supported by transparent international dialogue involving not just scientists and developers, but ethicists, policymakers, economists, environmental groups, and the public.
Everyone needs to be involved.
Absolutely. Only through such concerted, collaborative efforts can we hope to ensure that CRISPR's incredible power is harnessed responsibly and its immense environmental dangers are meticulously managed, preventing irreversible harm to both ecosystems and human societies.
Okay, so wrapping things up. CRISPR truly stands as a monument to scientific ingenuity, a testament to our ability to understand and then repurpose nature's elegant mechanisms for profound human benefit. We've seen its immense power today.
Undeniable power.
From the potential to offer a one-time cure for devastating genetic diseases with therapies like Cas9 to revolutionizing cancer treatment and even addressing global challenges in food security and environmental resilience through sustainable agriculture.
But as we've explored in detail, its very strengths also give rise to inherent dangers. The technical peril of unintended mutations and broader genomic instability, though actively being addressed by next-generation platforms like base and prime editing.
Which offer much hope.
They do. But the risks remain a critical consideration, particularly in clinical applications. Moreover, the ethical and societal dilemmas surrounding its use in humans and the environment are deep and unresolved.
Yeah, the germline debate.
The debate on human germline editing. The risks of social inequality exacerbated by high costs and access issues, and the biosecurity threats posed by powerful tools like gene drives, all underscore the urgent need for a cautious, thoughtful, and globally coordinated approach.
The path forward for CRISPR, then. It isn't about blind embrace or outright prohibition. It seems it has to be a path of responsible innovation,
That's the goal.
requiring a collective commitment to international collaboration, transparent regulatory frameworks, and inclusive public deliberation. So we leave you, our listener, with this final thought. What do you think is the most critical next step for society in navigating this delicate balance between CRISPR's incredible power and its profound perils?
And perhaps, how do we ensure that this incredible power, this ability to literally edit life itself, is harnessed not just for the benefit of a privileged few or for narrow interests, but for the equitable advancement and well-being of all humanity? The future of life editing, and indeed perhaps the future of our species, truly rests on our collective ability to thoughtfully and ethically answer that very complex question.